Hello, dear students. Today we are starting a new chapter that is your principles of inheritance and variation. This is revision short one. We are doing revisions for CBSE exam, upcoming exam 2026. Terminology and Mendel's laws. We'll be discussing about terminology in genetics that's used there. So number one is inheritance. Inheritance means passing of traits from parent to offspring. Number two is your hybridization. Hybridization means crossing two different plants. Then true breeding varieties, plants that show the same trait generation after generation. Then what is a trait? Trait is actually a specific feature. Like when we talk about height, height is maybe tall or it may be dwarf. So tall and dwarf is a trait. When we talk about character, character is a general feature like height. Height is a general feature, and this character has traits. It may be tall, it may be dwarf. Then alleles, different forms of the same gene. Like if we are talking about tall, like capital T, small t. These are two alleles. We are talking about tall, capital T, small t. This is tall, hybrid tall. So these are two alleles of the tall, capital T, small t. Then gene is a unit of inheritance controlling a trait. Then you have dominant. Dominant means trait that appears in F1 generation. Recessive trait that is hidden in F1 generation but appears in F2 generation. Homozygous means same alleles like capital T, this is same, small t, this is same, so they are homozygous. Heterozygous means capital T, small t. They're different. Then genotype means genetic makeup, whether it is tall is capital T, or it is capital T, small t. So we are checking here we are checking genes. What type of genes are making that particular phenotype or genotype? Then phenotype means physical appearance. Like if it is capital T, it is tall. It is small t, capital T, it is tall. So that means phenotype, external appearance, how it appears, whether it appears tall or dwarf. Then you have segregation. Segregation means separation of alleles during gamete formation. Like you have capital T, where crossing with capital T, or small t. Then these alleles will separate first. Capital T, and this small t, small t. This is called segregation process. Then you have Punnett scale. Punnett scale is that box diagram to predict the offsprings. Then F1 generation. F1 generation is first offspring generation. F2 generation is the offsprings from the selfing of F1 generation. Then gametes are sex cells carrying one allele. So this is all the terminology that is there in genetics. In this part of the genetics, that is principles of inheritance and variation. Now, why did Mendel choose the pea plant? Number one reason is easy to grow. It was easy to grow. It was short. It was having short life cycle. And there were clear contrasting traits like tall, dwarf. Then self-pollination was possible and large number of offsprings were formed. Then key terminology we are done. Mendel's laws of inheritance. Number one law is your law of segregation or law of segregation we'll talk about. So law of segregation states alleles separate during gamete formation. Each gamete carries only one allele. You have capital T, small t. So during gamete formation, they will segregate from each other. That is your law of segregation. This explains the appearance of recessive traits in F2 generation. Okay, this is law of segregation is explaining this thing to us. Then you have law of dominance. Dominant allele shows effect even in the heterozygous condition, whether it is capital T, small t. So even in heterozygous condition, it shows its effect. Recessive allele expresses only when it is homozygous. That means small t, small t. So when we do the crossing of capital T with small t, small t. So F1 generation is capital T, small t. So we have a dominant trait. So this dominance tells us that this dominant trait expresses itself in F1 generation. Then why is there no blending? Tall and dwarf, they remain separate. There is no medium height. Traits reappear unchanged. Then when we talk about this F1 generation observation and F2 generation, in F1 generation, like if we are talking about the crossing between capital T, small t, it will be dominant. So F2 generation, the ratios are very important. If we are taking this cross, and the phenotypic ratio will be 3:1. 3 will be tall, 1 will be dwarf, and genotypic ratio will be 1:2. 2 is to 1. One will be pure tall, two will be heterozygous tall, and one will be pure dwarf. So these ratios are very important. You must remember them. Then Punnett square diagram you already know, and then test cross and its definition. So this test cross thing is very, very important. You actually, by doing this test cross, you determine the genotype of parents. What was the genotype of parents? Like for example, if you have this in like F2 generation, if all are tall, if you get all tall in F2 generation, that means it will indicate that the parent was capital T, capital T. The parent was capital T, capital T. If the ratio is 2 is to 2 or 1 is to 1, you can say, that means parent is capital T, small t. So this is how you determine the parental genotype when you do the test cross. It's very important. So a test cross is a cross between an organism with dominant phenotype and a homozygous recessive organism to determine the genotype. So these are important things that are at the beginning of this particular chapter. So now we'll be discussing about this incomplete dominance after this. Thank you very much for watching this video.